All right, so far what we've seen is transcription. We've seen how the different RNAs are made. We looked specifically at mRNA, tRNA, and ribosomal RNA so far. Now we're going to start to get into translation. But one of the things we forget a lot of times is that when we go from transcription to translation, number one, they're not even in the same biochemical compartment. First of all, transcription has to occur where the DNA is, so in eukaryotes it's in the nucleus, and translation is in the cytosol, or cytoplasm. And I suppose you could say there's also ribosomes on the endoplasmic reticulum, rough ER, but they're certainly not in the nucleus. So we're going to have to get the mRNA and all the other RNAs out of the nucleus into the cytoplasm. And in doing so, there's an intermediate step that's often neglected. We know hopefully by now that transfer RNAs, or tRNAs, are the RNA moieties that physically bind and carry amino acids to the ribosome. Okay, this is sort of a setup of what we're going to have in the ribosome. So here's the mRNA that's going to run from 5' to 3'. Hopefully we know that the mRNA possesses a codon, which is a sequence of 3 nucleotides. In this case it's AUC, and in order from 5' to 3', if we talk about the nucleotides in that codon, we talk about it 1, 2, 3, labeled from 5' to 3'. Now, in every sense of the word, the tRNA is going to be anti-parallel to the mRNA. Now, the tRNA has a very strange looking secondary or tertiary structure, depending on how you want to look at it. But in any case, if the mRNA runs 5' to 3', even though we have the secondary structure, the tRNA runs 3' to 5'. So if we're talking about the codon going, we'll say from left to right here, the codon is 1, 2, 3, then the anticodon is listed 3, 2, 1, because we have to name from 1 to 3 the tRNA anticodon from 5' to 3'. However, the way we normally look at it is that the mRNA is going to be anti-parallel to the tRNA, meaning the mRNA codon is anti-parallel to the tRNA anticodon. So, if the codon is AUC, we want it to be complementary, so the anticodon will be UAG. Now, on the 3' end right here, there would have been a 3' OH group, but through the action of an enzyme that we're about to look at, that OH group on the 3' end gets ligated to an amino acid. And so, by the codon interacting with this anticodon of the tRNA, you have this amino acid here on the 3' end. So the codon literally, by this interaction, facilitates the bringing in a specific amino acid right here. Okay, if we had a different codon right here, there'd be a different anticodon, a different tRNA, and a different amino acid. And we'll look at that in more detail. But basically what we're saying is that the codon dictates the anticodon, which dictates the tRNA, which dictates the amino acid. That's how a codon codes for a specific amino acid. All right, and these are hydrogen bond electrostatic interactions. We'll talk about this a little bit later, but this third position on the codon, which by definition corresponds to the first position on the anticodon, it's sort of you have to basically figure out ultimately the perspective that you're going to talk about it with. But ultimately the third position on the codon is sometimes called the wobble position, because remember that the anticodon, and particularly the tRNA, can have what we call funny bases. Remember those modified bases that we previously looked at where we can make different bases? Here's a picture of some of them. Well, it turns out due to those modified bases and a little bit of extra flexibility here, this base right here that's position 1 on the anticodon can have different identities and not be exactly complementary to position 3 on the codon. That's called the wobble base, and it leads to some interesting things like you can have more than one tRNA binding to this codon. Okay? Now, protein synthesis, when we're talking about the ribosome, requires something called an aminoacyl tRNA. What is an aminoacyl tRNA? Remember back to when we looked at the tRNA structure. And by the way, here is the anticodon arm of the tRNA. This is where the anticodon is. Remember on the 5' side of the anticodon, that's the wobble position. Okay, so here's the 5' end, wobble position. That's position 1 on the anticodon. Up here at the top of the tRNA, the 3' end, we have the amino acid arm. The 3' hydroxyl of this adenosine up here at the top, at the 3' end, is going to covalently bind an amino acid. Now, this is actually what the tRNA more or less looks like. It doesn't actually look like this. This is just sort of a diagram to help people understand where things are. If you actually looked at it according to this spacefill diagram, it actually looks more like this. Here's the anticodon right here in the yellow, and then up here at the 3' end, where my mouse is, that's where the amino acid binds. Now, how exactly does the amino acid bind? Well, when it binds, it looks like this. Now, the amino acid is going to be connected to that 3' hydroxyl in an ester fashion. So here's the rest of the tRNA. It's shrunk for obvious purposes. It's huge. And then here's the phosphate. This is the 5' phosphate of this adenosine. Here's the ribose, the adenine, and then here's the 3' oxygen, which is in an ester linkage to the amino acid. Okay? And the question is, how does that amino acid get there? Well, it's not just as simple as it just spontaneously does it. You have to have an enzyme to do it. And that enzyme is called aminoacyl tRNA synthetase. All right, so we're going to just look briefly at how it works. It turns out that the amino acids, remember amino acids have a carboxyl, an alpha carboxyl that is. The alpha carboxyl of the amino acid is going to attack the alpha phosphate of ATP, facilitating phosphoryl substitution, etc. You get loss of pyrophosphate, and now you have this, it's this adenylyl amino acid, or sometimes they call it a 5' aminoacyl adenylate. What it basically is it's an amino acid and an adenosine monophosphate in an ester linkage, basically. Sort of, you could think of it that way. It's an activated amino acid. Now, we have two classes of aminoacyl tRNA synthetases. All right, one of them, which is the class 1, is actually initially going to add the amino acid onto the 2' OH. The class 2 aminoacyl tRNA synthetases add it directly onto the 3'. So let's look at it first with respect to adding it onto the 3'. We'll look at the class 2 first. So the 3' OH is going to attack that ester carbon on the aminoacyl AMP. It's going to facilitate acyl substitution, loss of adenosine monophosphate, and you effectively now have the amino acid in an ester linkage to the ribose of the tRNA 3' end. Okay? That's a direct mechanism. The class 1 aminoacyl tRNA synthetases are a very indirect mechanism. In this case, the 2' OH is what's going to do the nucleophilic attack. It's going to attack in the same way, attack the ester carbon, facilitating acyl substitution and loss of adenosine monophosphate. However, now what you see is that we have the exact same linkage. We have an ester linkage between the amino acid and the ribose, except it's on the 2' position. So what has to happen in a follow-up step is what's called a transesterification, meaning we're going to switch the hydroxyl that is in the ester linkage to the amino acid. So the 3' OH now attacks that ester, acyl substitution, and now you effectively substitute this amino acid onto the 3'. So the net result of either one of these classes is the same. Okay, you get the amino acid in an ester linkage onto the 3' OH of the tRNA, and that 3' nucleotide happens to be an adenosine, but it just does so through a different mechanism. Now, remember, class 1 is the more indirect because you initially do the 2' and then transesterification. Class 2 aminoacyl tRNA synthetases are direct. They transfer the amino acid directly onto the 3' OH. Okay, what are the reasons for doing that? Not really sure. Okay, you look at the class 1 and class 2, it really appears through looking at a lot of different organisms and their tRNA synthetases that there are 10 amino acids that are in each group. Very symmetric, I suppose. You can look at these and there really doesn't seem to be a lot of reasoning. You might say, well, maybe it's based on the class of amino acid. Well, here's arginine, positive nitrogenous amino acid, lysine, positively charged nitrogenous. Here's tryptophan and tyrosine, two aromatics. Here's phenylalanine, the aromatic over here. Cysteine looks a lot like serine, but serine's in a different group. And I suppose you could say all the branched chain amino acids, isoleucine, valine, and leucine are all in this group. But the point is that there really doesn't appear to be much in the way of structure or class of amino acids that dictates which enzyme they use. And in fact, I don't really think they figured out why particular ones prefer one mechanism over the other. Okay, but these are the different classes. Class 1 is the indirect mechanism, class 2 is the direct mechanism. And that's basically how the amino acid gets put onto the tRNA. And so when this tRNA goes to the ribosome, this is how the amino acid is attached in this ester linkage to the ribose on the 3' OH. Now, because the amino acid is now in an ester linkage rather than a carboxyl, a simple carboxyl, we say the amino acid is activated. We often call this amino acid an activated amino acid, or we call it charging the tRNA. So sometimes when you hear somebody say charging the tRNA, what they mean is that you put an amino acid on it. Sometimes you'll hear it referred to as charging the amino acid. Okay, what it means is just activating it so that the ribosome can polymerize these amino acids. All right. So now that we've done that intermediate step and talked about tRNA synthetases, we can now start talking heavily about protein synthesis. So that's what we're going to do in this video, in the next video. All right, so make sure to like this and subscribe to the channel for future videos and notifications. Thank you.